Hello students, this is a short video about enzyme lab exercise number 5. This is an online lab and you'll be doing an at-home experiment in this lab. So let's take a look at the instruction document and then the worksheet so you know what you're doing. All right, let me see if I can get my computer to do the right thing here. And I want to go to the instruction document. I'm on the wrong page, but here we go. Exercise 5, enzymes. So we're going to learn about enzymes. What are they? How do they catalyze reactions? What factors affect enzyme activity? And there's a little bit of introductory material, so take your time reading that. There are 2 videos that we found online that are worth watching, and another Amoeba Sisters video, and another HHMI video on the evolution of lactose tolerance. So many of us can drink milk as adults, and that's because we still make an enzyme called lactase. And some of us can't drink milk without having an upset stomach. That's because you no longer make the enzyme lactase that helps to break down milk sugar, lactose. And this is an interesting video about the evolution of this kind of tolerance in humans. So check that out before you take the pre-lab quiz. And then there are some questions to think about. How do enzymes catalyze reactions? What are the optimal conditions for an enzyme, etc. And then you'll do take the pre-lab quiz, and then you'll do an at-home exercise. And this at-home exercise is called Stop the Browning. This at-home exercise has to do with an enzyme catalyzed reaction that causes our fruit to brown if we cut it and leave it on the counter for a while. So there's an enzyme called Polyphenol Oxidase that's found in the cells of a lot of fruit. And when it reacts with oxygen and another reactant, it produces a product that turns brown, that turns the fruit brown, and the fruit gets browner and browner over time as this reaction continues. We think that this is actually an adaptive reaction. If, say, a bruise happens on a growing fruit, that little bruise or cut could lead to microbes getting inside. And this reaction produces a product that is actually toxic to microorganisms. Then it also turns brown. The browning is one of the effects that we can see. It is not toxic, by the way, to us. Brown fruit are fine to eat. Okay, so it turns out that we do a lot of things to prevent this browning. If we want to have apples in a salad, for example, we put our apples in lemon juice before we treat them with lemon juice before we put them in the salad, so you can take them to the picnic in pre-COVID days and eat your apple salad with everyone else and not be kind of like brown apples, right? There are other treatments as well. Some other ideas include salt water or honey, apparently, or soda water, carbonated water. And what we're going to do at home is we're going to do an experiment to test 2 of these variables. Sorry, 2 of these possible treatments. And we're going to follow the directions to do our project, right? There is a video also that I've provided that you can link to here that will help you kind of walk you through the project. But I made a couple mistakes in the video, so make sure you're following the directions. But the basic idea is we're going to set up, we're going to cut a fruit into 12 slices and divide the experiment into 4 conditions, 2 control conditions and 2 treatment conditions. What we're going to do with these fruits are we're going to put the fruits, immerse the fruits that have been cut in a treatment for a few minutes, for 5 minutes, take them out, expose them to the air for an hour, take a picture, and look at what, have they turned brown or not, and then wait a day and take a picture again. So 2 treatments you get to pick. So you get to pick, maybe I'll try honey, or maybe I'll try salt water, or maybe I'll try lemon juice. And I'll submerse submerse my fruit into that lemon juice for 5 minutes or into the salt water for 5 minutes. And then we have two treatments that are controls. We have a treatment of water, just plain water, as one of our controls. This is a positive control. We would expect, if you put the fruit in water for 5 minutes, water isn't going to help it to prevent it from browning in any way. So I would expect the fruit to still brown up. And we have a negative control, in which we take the fruit and we immediately put the fruit into a baggie with no air, with no oxygen. The fruit should not undergo this browning reaction. The enzyme catalyzed reaction requires oxygen. So if we cut the fruit, put it quickly in the baggie, and leave it in the baggie sealed up, squeezing out all the extra air from that particular control baggie, then we probably won't see much browning in that fruit. We're going to have to find out. You get to pick the fruit. Pick something that you think will brown. Potatoes brown, peaches brown, some apples brown better than others, bananas, avocados get gross, right? So check out this experiment. You're going to be doing this experiment. You can watch the other video if you want, following the directions. This will take you a day, and you'll need to acquire these materials to get them, right? And then you're going to address all the questions on your worksheet. So we could click on that worksheet. I'm going to head over and check it out. And on this worksheet, you see stop the browning. That's what we've just done with our fruit or vegetable. There are some questions about controls, the two treatments you're choosing to use, and conclusions. What did you find? All right, compare your treatments to the two controls. And also, we want you to take some pictures because it'll help you make those comparisons as well, especially getting a before and an after, if you will. An initial picture treatment, before treatment, 1 hour after treatment, and then 24 hours after treatment for all 4 conditions. All right, so we got the fruit in the baggie, the no air control, the fruit in water, the negative control, the fruit in your 2 treatments. All right? That's the first part of the experiment. The second part of the experiment, if we go back to our document, is another online simulation. In this simulation, you're going to be looking at how an enzyme reaction proceeds under different conditions. And we're looking at an enzyme simulation that is on one of those same sites we looked at last time, John Darkow's Enzyme Simulation site. So I'm going to click on that and just walk you through it a little bit. Enzymes. The simulation that we're going to be doing has some background information on it, so you might want to read that to understand it better. And then it has a simulate tab where we can actually do some simulations and run them. And the enzyme catalyzed reaction that we're going to look at is the digestion of lactose. Lactose is milk sugar. It's made up of glucose and galactose linked together with a covalent bond. This is chapter 5 material, people, and from lecture. And lactase is an enzyme that can catalyze the breaking of lactose into its 2 monomers. Okay? Lactase is functions in different kinds of conditions, and we might predict that lactase would function best where it usually does its job of breaking down lactose. So the question is, where does that happen? Does lactase typically work in the stomach at a pH of 2, or does it do its job in the small intestine at a pH of 6 or 7? Well, we're going to find out. So in this simulation, you can modify a whole bunch of different things. So you start with an initial concentration. We're going to leave that the same, of 48 micromolarity of lactose. All right, that's the sugar we need to break down. And we're going to use the same temperature. 25 degrees, it's like a little warmer than room temperature, 25 degrees Celsius. And the pH, that's the acidity of the environment. And this could have a huge effect. We are going to actually modify pH. That's what our experiment is all about. And then the sample size, we're going to do a sample size of 20 because we want to do it several times to see kind of an average effect. And we're going to use lactase enzyme. In the presence of lactase, we want to see what this enzyme catalyzed reaction looks like with the enzyme. All right. The thing that we're going to measure, guys, is the glucose production. Actually, I think it's going to be yes, I think we're supposed to do the mean of samples. Let me double check. I better go back and read what we're supposed to be looking at here. So I'm scrolling through here. No, we're looking at glucose production, I guess. Okay, let's go back to my simulation and click on glucose production. How much glucose we make. Why are we looking at glucose to see how fast this reaction goes? Remember, in the reaction, lactose is broken up into glucose and galactose. So one of the products is glucose. The more that this reaction proceeds, the more glucose we should get. So if the enzyme is working well, then we should get a lot of glucose. If the enzyme is not working well, we shouldn't get very much glucose. Okay? Okay, and so what we're going to do is we're going to run this experiment at a pH of 7 just to see what happens, to see if we get much glucose production. And let's give it a try. I click on run. Whoa. Maybe I need to find the mean of samples. Oh, that's better. It is the mean of samples we're looking for. So over time, over these 40 minutes of time, I found an average glucose concentration of about 46 or 47 micromolarity. Okay, great. What about if I change the pH? And in your job is to change the pH 15 times and record the average glucose concentration. And we can go back and take a look at that information a minute. Down here, we get this information. Let's see. Figure out what the glucose, okay, so blah, 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 change the lactose concentration. Make sure that you're changing things to the mean of samples. We got that going. We got the lactose concentration correct, the temperature correct. We got the lactase correct. We are ready to go, except it says here, figure out what the mean glucose concentration is at 20 minutes. I'm going to make that 40 minutes if, in fact, our, I think that's wrong. I think it should be 40 minutes because that's what it looks like on our simulation, doesn't it? Let's go back. And so you need to then run this. Yes, in fact, this is our time over 40 minutes. Let's do 40 minutes. I'm not sure why we chose 20 minutes. Let's do 40 minutes. We're going to say, yes, after 40 minutes, the average glucose concentration is 46 micromolarity under a pH of 7. Now I'm going to reset this and change my pH and vary it. You get to choose which numbers you use, but you need to use a range of pH samples all the way from 0 to 14. You have to choose 15 different samples and record the average glucose concentration for those 15 different runs. And so I'm going to run it under a pH of 0. Ooh, in a very acidic environment, yikers, we don't get any glucose produced. Lactase does not work at a pH of 0. I'm just going to try a couple others just to let you see. What about a pH of 4? Pretty acidic. Okay, so here, lactose does run pretty well. The final average glucose concentration was 33, all right? 33 micromolarity. Let's try a pH of 6 and see what happens. Oh, that worked really well. So you're going to record these, and then we'll go to our Google Doc again. After you record 15 different pH 15 different data points for the 15 different pH settings you've chosen, you're now ready to make a graph. So if I click on my worksheet assignment again, I can see that I'm down here looking at changing the conditions at my online simulation, and I will be making a title for my table, and I will be writing in the different pHs I've chosen, as well as the glucose concentration at 20 minutes. Let's change that to 40 minutes, people, because that's what the presentation looks at, or the simulation looks at. And then you're going to be making a graph of your results. So you're going to graph the effect of pH on the rate of this reaction, or on the concentration of glucose. So glucose, the concentration of glucose, is your dependent variable, the one on the y-axis. The pH is the independent variable, the one that you've manipulated or changed. And you make a graph. Oh, but wait a minute, we're going to make an Excel graph this time. So if you've never made a graph in Excel, there's an instructional video, how to make a good graph in Excel. And you're going to make a graph in Excel that looks at the effects of pH on the rate of this reaction. And then you have to write a figure caption. So once you get your graph made, you copy and paste it, put it into this document, make a good caption. I've given you all sorts of information about what to include in a good caption, all right? And then draw a conclusion. And here, we want to know what's the best pH for lactase to work. Where does it work the best? We call that the optimal pH. And you're going to sort that out and tell me, looking at your graph, making some conclusions based on your graph. I think that's all we need to know for exercise 5. I hope it goes well for you, and that'll be it, I think. Thanks.